Welcome back everyone. Right now I'm in the Shellfish Genetics Lab at the Pearl, the PATUXENT Environmental Aquatic Research Lab. So if you watch my second and third video, those are the things that I was doing as an intern while I was here helping out in the hatchery. But now since your girl is a recent graduate, taking a gap year, I have time on my hands, skills to learn, and a research project to finish. So now I'm an employee at the Pearl. The reason why we're in the lab today is to continue my project this past summer. It is titled Using PCR Assay to Detect Dermal Disease in Eastern Oysters in Maryland waterways. First, we need to know what is dermal disease and why is this research important. Dermal disease, its scientific name is Parkinsis marinus, is a protozoan endoparasite that is responsible for mass mortalities in the eastern oyster. Dermal disease is a waterborne disease. If I had a freshly shucked oyster, this demonstration would be a lot easier if I could show the anatomy, but I'll probably just put a picture or a video as I'm explaining. So this is a half shell of an oyster, and then the gills are found in this area right here. So once an oyster ingests the disease, it goes through the gills and then it continues throughout the anatomy of the oyster, causing tissue lysis and then ultimately killing the oyster. So for example, let's say this is the infected guy, it ingested the disease and it's proliferating throughout it. So the dermal requires the oyster's body to reproduce. So it's going through this process, this infected oyster, then we have an uninfected oyster right next to it. So once the disease proliferates throughout it and ultimately kills this oyster, it will travel through the water and then is ingested by the gills of this uninfected oyster, which is now infected. High intensity infections and high mortality rates, they occur in the warmer water temperatures, so in the summertime, as well as having high salinity water ranging between 12 parts per thousand to 15 parts per thousand. So the infection period isn't long, it's not year round, but it is short, and during that time mass mortalities does occur. And the interesting thing about this disease is, let's say it's infected an oyster and it's began its proliferation process but has not completed it. And when it gets a little bit colder, when the salinity of the water is lower, it will become dormant, it'll be stagnant until it gets warmer again, and then it'll continue or resume the proliferation process, which makes this disease a nuisance. So first, after we extract the gills from all the oysters from the different locations, we take a very small piece of it and put it in here in the centrifuge tube. You can barely see it, and we do the DNA extraction process, which is just separating the DNA from the other macromolecules. After the DNA extraction, where the macromolecules are separated from the DNA, I put it in a centrifuge for 15 minutes to further the separation, and then I refrigerated it for 2 hours, just so it gets a chance, just to sit. Next is the PCR, the polymerase chain reaction process. And this process involves 3 steps. Step 1 is denaturing. So the DNA goes from double-stranded to single-stranded. Next is the annealing process, where the primer... Alright, that was a hornet, flying around, and it's attracted to light, so I'm turning the light off so it's not flying around or stay on the wall or something. So yes, next, PCR. For the denaturing process, the DNA is converted or unwinded from a double-stranded to a single-stranded DNA under the right temperature conditions. Next is the annealing process, where the primer will attach to the DNA of interest. So believe it or not, in the DNA extract, you're not only going to find oyster DNA and dermal disease DNA. If the oyster is infected with dermal, there's a bunch of other DNA that's in that as well. So we need to be specific about which DNA we're trying to target. That's why a primer, a short nucleotide sequence, is required, and the primer of dermal is known and we do have it. So yes, annealing. The third step is extension. So the primer is attached to the DNA of interest, and now it just needs to replicate, and then just continue to replicate and just keep that cycle going. So for the next step, we need a PCR machine. We do have a PCR machine, and then we need the required reagents. First, we have the PCR buffer. I feel like this is a makeup video. PCR buffer. Next, we have the DNTP, deoxynucleotide solution mix. This contains the base pairs for the extension stage process. Next, we need the forward and the reverse primer. When I was doing this project over the summer, we had the incorrect forward primer. There was a mistake in the literature, but now we have the correct one. Next is TAC polymerase. And this is responsible for helping the DNTP, the base pairs in the DNTP to connect to the primer to execute extension. So then these have a protocol where we mix them all together. They have their specific amounts in microliters, so you know we're gonna need some pipettes for real. And here we have it. Here's our mixture. I did forget that we do add distilled water here, but don't drink it. Yes. Now to prepare to actually go into the PCR machine, I'm gonna put 20 microliters of the assay. I think that's the right term. Using this, using this pipette into here, and then 5 microliters of the DNA template. This is our lovely T100 thermal cycler, our PCR machine. The sample has been in here for 1 hour and 51 minutes. The run has been completed. Cancel, are you sure? Yes. The run is completed. I didn't cancel, I just wanted it to stop. Boom. Here it is. Oh, here it is. Throw it in this tray right here. On to the next step. So we've done the DNA extraction process, we've done the PCR process. Next is the gel electrophoresis. So, gel electrophoresis is a method used to separate DNA by its molecular size by way of electrical field charge. We have everything loaded up the gel, and it is running. Those bubbles is electricity. We have it running on 100 volts for at least 45 minutes to an hour. So since DNA is negatively charged, we're starting on this end, and it is going to obviously attract the opposite, so that's why it's pulling to the positive end. And you'll see what that looks like in a little bit. Okay, now it's been running for an hour, so we're gonna stop it, remove the top. I don't want to get electrocuted. I forgot something. I've got it. This is what we're gonna slide our gel onto. Oh my gosh, it's floating away. Oh my gosh, it's floating away. Oh man. Be gentle with the gel. Get that liquid off. Slide it under here. Alright, so the next thing I'm gonna do is look at this under a blue light. So now for the final step, we'll be able to examine the gel under the blue light. So if there is dermo in one of the DNA samples, then we will see a bright green strand towards the middle of the gel, or just like where it ended on the gel. Fingers crossed there's no bright strands there. But what I always do is I add a negative control and a positive control. My mentor said I don't really need a negative control since we know that this is the correct primer, but I just do it just in case, you know, because you never really know. So on the gel, I'm supposed to see a strand for the positive control. I'm not supposed to see one for the negative control. And then with the other samples, it's whatever, if it's infected, if it's not. So let's see. Well, unfortunately, there is nothing on the gel. And this is what heartbreak looks like, where nothing comes up on the gel. So you're here asking yourself questions on what did you do wrong. Not even a positive control strand. But you know, that's what research is about, you know, trial and error and figuring out what went wrong. So it is 7 o'clock at night. I'm gonna go home, and then tomorrow we're gonna try this again. You know, I've been here all day. This is a long process. You know, science.